And they really feel how they Oh, no, I voted like every one. Essentially. Including anyway, maybe I can just show you like what obviously like the distribution is
We need to get started, so we're going to run over by a few minutes. We're going to try to pack 3 talks into this hour period. So apologies for anybody has to leave. If you need to leave sort of partway through, probably about 25 to 1, we're going to start Ken's talk. So I'm going to go, then Jack's going to step in, and then Ken will do his. All right, so we all ready? Okay, so thank you all for coming. My name is Brien Riley, and I'm going to describe for you the molecular genetics projects that have been underway at the FABG over the last 10 years. So our institute has a long history of molecular genetics work, founded on a number of major sample collections, families, case controls, and twins ascertained for a number of phenotypes, including schizophrenia, alcohol dependence, major depression, and anxiety disorders, all of which are going to be covered today, as well as nicotine dependence and post-traumatic stress disorder. In total, our samples number about 75,000, and with our collaborative networks, this increases to about 200,000. Since 2006, our focus has been on genome-wide association studies and sequencing, and I'm going to give you a summary of 3 major molecular projects from 2008 to 2015 and our current directions, and then I'm going to hand over to Dr. Jack Hedema to talk about his genome-wide association study work with anxiety disorders. So the first study, both chronologically and to be discussed today, is our GWAS of schizophrenia. Together with 2 research groups in Ireland, we initiated the Irish Schizophrenia Genomics Consortium in 2007. This is an international collaboration involving researchers at VCU, Trinity College Dublin, and Queen's University Belfast. And we also acquired international funding both from the Wellcome Trust Case Control Consortium 2 and NIMH. This sample includes 1,600 cases and just under 1,800 population controls, all from Ireland or Northern Ireland, and reporting all 4 grandparents born in Ireland or the UK. This is to try to minimize ethnic and genetic heterogeneity within the sample. Our replication was undertaken in 13,000 cases and 31,000 controls from the independent samples within the PGC1, the SGene Plus Decode Consortium, and additional samples from the Wellcome Trust Case Control Consortium. Our data were included in both the PGC1 and PGC2 schizophrenia cohorts, and this study has yielded 33 publications so far as of today. So, as you might expect, in a sample of this size, we detect 1 genome-wide significant signal here in the MHC region on chromosome 6. This RS204999 had a p-value of about 3 × 10⁻⁸. And when we look at replication, this particular SNP, which is our strongest discovery signal, is at about 32 megabases on chromosome 6. There are 2 semi-independent peaks within the MHC region that are regularly detected in schizophrenia. One of them at 32 megabases, and the more prominent one here at 30 megabases. So when we go to the replication sample, the combined p-value for the 32 megabase position, our strongest discovery SNP, does increase somewhat to 1 × 10⁻⁹. But overall, the stronger signal in the combined sample is at this 30 megabase locus, where the most prominent signal is seen in the PGC analysis. Our most important novel discovery in this study was the identification of duplications partially overlapping the PAX7 gene on chromosome 20 in the discovery sample, plus additional replication cases of schizophrenia and bipolar disorder and controls from Ireland and the UK. We observed 22 copies of this duplication in about 12,000 cases, and 10 copies of the duplication in about 21,000 controls, for a p-value of 10 to the -4 and a pretty strong odds ratio of about 11.3. The most interesting feature of this CNV is that the allele itself, the duplication allele itself, is in very strong linkage disequilibrium with local haplotypes. This is a very, very significant feedback. And what this suggests is that unlike most copy number variants, which are recurrent events that happen on different chromosomal backgrounds in different individuals, this is indicative of a single ancestral event. So this duplication is actually being inherited in all of these individuals identical by descent from a single common ancestor. That's very unusual for copy number variants, and we don't really see this outside of the British Isles population yet so far. So this would appear to be limited to the far extremes of northwestern Europe. So our data for schizophrenia was included in the PGC2 2014 mega-analysis. This was a study of just over 80,000 individuals in total that identified 108 genome-wide significant and independent sites for schizophrenia. So this is the current genomic landscape for genetic risk of schizophrenia. And this was an incredibly important step. This was the largest genetic study of schizophrenia ever performed, and this is by far the most successful and most significant set of genetic associations identified yet. However, there are some remaining issues here, and despite this success, we do see some clustering of signals in genes related to glutamate neurotransmission, genes related to synaptic plasticity, and genes coding calcium channel subunits. However, only 10 of the signals are attributable to non-synonymous SNPs, things that might actually be expected to be functional alleles in the genome. And we do see a pretty strong degree of enrichment in functional non-coding elements, so outside of the exons and protein coding sequences in genes. In regulatory elements, in the sites of chemical modification, both methylation and acetylation, and in other sequences that are conserved between species but are of currently unknown function. The real challenge with this data is that, but there are two challenges. First, these signals can be driven by hundreds, or even in the case of this MHC signal on chromosome 6, thousands of SNPs, all in very strong LD with each other. So trying to parse out that SNP set and identify the specific single variant that's actually driving the signal, the thing that's really causing risk in cases compared to controls, is extremely challenging because of linkage disequilibrium. And even if we could identify the key functional SNP from this set of several hundred or several thousand that are driving the signals, the effect sizes for these alleles are very, very, very small, tiny increases in risk. So even if we could identify the functional allele, there's no guarantee that we're going to be able to detect an effect of that allele in some kind of downstream molecular biological or behavioral analysis. So we're kind of a little bit stuck here with moving forward with the common variant data. So a number of people, ourselves included, are also seeking to perform sequencing either at the exome or whole genome level on our available samples for whom we have GWAS data, to add in rare variant analyses in conjunction with common variant analyses. This is just showing you the results of some work we've been doing in the past year. There's an interesting feature in the genetic epidemiology of schizophrenia, which is that despite the fact that heritability for schizophrenia is consistently estimated at 70 to 80%, and despite the fact that risk in the first-degree relatives of index cases is substantially elevated, so on average, a first-degree relative of the schizophrenic case has 10 times the population baseline risk for the disease. Despite that incredibly consistent and very strong evidence, if you look at serial singleton cases sampled from the population and ask them about family history, in general, only a quarter to a third of such cases report any family history whatsoever. And 2/3 to 3/4 of samples report no family history. So what that means is that when you look at multiplex families, like are represented in the center portion of this graph here, where you have multiple cases within a single SIB ship, or multiple cases within both the first and second degree relatives within the family, these families have substantially higher recurrence risk than is indicated for singleton cases in the population because of the much higher number of affected individuals in these families. So anyone who's spent a lot of time working with GWAS data over the last few years would probably have the same initial thought. If we're going to try to explain this elevated recurrence risk, how can we do so? And the first test to perform, I think always, is to ask whether the overall loading of common variant risk factors in the families is higher than it is in the singleton cases. That is, to do the polygenic risk scores, are polygenic risk scores on average higher in family members than they are in the singleton cases for the population? So we undertook that analysis, and what this graph is showing you is that the answer to that question is no. Family members do not have elevated polygenic risk scores compared to singleton cases. So here's the value for the singleton cases from Ireland. These are the core schizophrenia cases from the multiplex families. These are the narrow spectrum psychosis cases from the families, the broad spectrum cases from the families, and even here, these are the individuals who have no psychiatric phenotype whatsoever, the unaffected members of these multiplex families. None of these groups are significantly different from the polygenic risk score in the singleton cases. All 5 of these groups are significantly elevated over the polygenic risk score in controls. So we cannot explain the elevated recurrence risk on the basis of excess common variant loading. And so the natural second hypothesis would be that these families are segregating either rare, higher impact simple nucleotide variation, individual base changes or small indels, or rare larger copy number variations that substantially increase their risk. And we've started to undertake some sequencing work in the families. We collected whole exome data on 78 cases. These are all affected individuals, pairs of affected individuals from 39 of our families. And we put the variants that were identified through a relatively primitive weighting scheme. And we identified one really standout variant in this NAA16 gene, driven primarily by the CAD score. This is a measure of the impact of the change on protein function. So this score value suggests that this change, the non-reference allele, has a significant impact on the function of this protein. The Philo-P score indicates a very high degree of conservation across multiple species and many millions of years of evolution. And most strikingly is the case to control minor allele frequency ratio. In the members of our families, after correction for genetic relatedness, this allele has a frequency of about 2%. In nearly 2,400 controls from the UK 10K sample, this allele has a frequency of 1 in 10,000. So it's 20 times as frequent in the members of our multiplex schizophrenia families as it is in members of the general population. This is a missense change, so it is actually a functional allele in this N-alpha acetyltransferase 16 gene. This is part of a protein complex that adds acetyl groups to polypeptides as they emerge from translation on the ribosome. And de novo copy number variants overlapping this gene have previously been reported in schizophrenia. So we already have one piece of data indicating that highly functional changes in this gene can increase risk for schizophrenia. We then went back to the PGC2 data to ask whether there's any evidence of association there. And we do see modest, this is nowhere near strong enough to achieve genome-wide significance in the current data set, but there is clearly a haplotype which is of elevated frequency in cases compared to controls within the large PGC2 sample. It would be very, very useful to know what the PGC result for this marker was, but unfortunately, because it's got relatively low frequency and therefore also has low imputation information, it was filtered from the PGC2 data set post-imputation. It would also be interesting to go back to our data and ask if we could reconstruct the haplotype that this allele is on, but haplotypic reconstruction in the dosage data in the PGC is virtually impossible, so we can't really make any more forward progress there. And this is really just meant to serve as a kind of proof of principle that sequencing members of these multiplex families can in fact identify alleles for you that do seem to have pretty strong impact on risk and are supported by other prior data. Okay, so now I'm going to move on to our GWAS of alcohol dependence. This was undertaken in the context of the VCU Alcohol Research Center. This sample included 706 probands and affected siblings. This is a relatively modest sample for such a complicated phenotype. But you do increase your power and decrease your noise slightly by genotyping relative pairs and then analyzing them appropriately for their relatedness. So for analysis, we use the modified quasi-likelihood score, or MQLS, which includes a genetic relationship matrix to account for the relatedness of the probands and siblings. We analyzed the affected individuals with the existing control data set from the schizophrenia study. This number is slightly different than on the earlier slide because we recalled all of the GWAS data from our Irish samples independently after the Wellman Trust-based control analysis. Now, these are population controls, which means that they are very, very lightly screened. Essentially, these are blood donors at the Irish National Blood Transfusion Service. They were asked if they had ever had any problems with alcohol or any history of psychotic illness, and if they denied both of those, they were accepted as a control. So very lightly screened. For schizophrenia with a 1% prevalence in the population, lack of screening in your controls has a really minimal impact on the power of your analysis. But for alcohol dependence, which is between 10 and 20% prevalence in the general population, that has an enormous impact on your power. So to try to deal with the fact that the controls were not screened for this much higher prevalence phenotype, we estimated alcohol dependence prevalence in both population samples using the NESARC data and in unpaid blood donors from Holland, who seemed like the most similar group of people we could identify to these controlled blood donors we were trying to provide an estimate for. And we used this estimate of 8.9% in analysis to account for the lack of control screening. Replication was undertaken in about 15,000 subjects of European descent from the Koga, German, and Yale UPenn case control samples and the Australian population twin sample from QIMR. The most important aspect of this study, however, was our access to model organism follow-up studies through our partners in the VCU ARC. So in this study, we detect 2 genome-wide significant signals. I will only talk about this one briefly because there appears to be an issue, an anomaly in the data with this, but I'll describe this signal and a couple of others in some detail. Our strongest signal is on chromosome 2 in the middle of the collagen 6A3 gene. This is an alpha chain of collagen 6. It's part of the extracellular matrix, and this particular extracellular matrix protein is brain expressed. Now, collagens in general might seem like odd candidates for a substance use phenotype, but there is actually a really large literature on the importance of both the components of the extracellular matrix and the capacity of cells to remodel that extracellular matrix for the neuroplastic changes that are observed following exposure to a substance. So, for example, if you block remodeling of the extracellular matrix, you block the expected neuroplastic changes seen in response to a single treatment of alcohol, of methamphetamine, and of opiates. So these are actually quite good candidates for substance phenotypes. Also of very strong interest, as will become clear in a moment, the mouse syntenic region has a very strong QTL for a phenotype called handling-induced convulsions. So this is, you give the mouse a single, usually intraperitoneal injection of ethanol. You wait a specified period of time, somewhere between 20 and 30 minutes, and then you pick the mouse up by the tail. Many mice will go into a spontaneous seizure when they're picked up by the tail in this paradigm. And although many people will describe this as a measure of withdrawal, because this phenotype occurs in a single trial, a single exposure trial to alcohol, and occurs temporally very close to the initial exposure to the alcohol, this is really better understood as a measure of initial sensitivity. And that's going to be phenotypically quite important in just a second. Okay, so the bulk of our model organism work was done in the C. elegans lab run by Jill Benninger and Andrew Davies, and in Mike Miles' mouse lab. I'm going to have to explain what's going on in this figure briefly so that you understand what's being shown here. In order to test the impact of manipulating a gene on behavioral response to ethanol in C. elegans, what we do is first we identify a wild type strain And then in this particular case, there was no mutant available for the best ortholog of Cal6A3 in C. elegans. So instead of a mutant, we used an RNAi knockdown approach for this study. So the manipulated animals are always shown in black symbols, and the wild type always shown in white. We collect baseline measures on both strains, measuring the speed of locomotion on an agar plate with no alcohol contained in it, and that's our baseline measure. And then the animals are exposed to a second agar plate, either with 200 or 400 millimolar ethanol in the agarose. And as they move across this surface, they begin to absorb alcohol. And what we're measuring here is at 10 minutes, we take a measure of their speed, and we relate that measure of their speed to their baseline. So what you're seeing here on the x-axis is the percent of their baseline speed that they maintain after 10 minutes on the alcohol plate. And what this shows is that the knockdown animals maintain about 40% of their baseline speed, whereas the wild-type animals maintain about 30% of their baseline speed. So the knockdown animals are less sensitive to the initial exposure to alcohol than wild-type animals are. When we look at mouse brain gene expression data, we see that Cal6A3 expression is positively correlated with handling-induced convulsions, which is a measure of sensitivity. So what these two sets of data are showing you is that if you knock down the expression of Cal6A3, you reduce initial sensitivity, and if you increase the expression of Cal6A3, you increase sensitivity. So these are really beautifully correlated phenotypes from an invertebrate nematode and the mammal, suggesting that the impact of this gene on response to alcohol is similar across species. We get a nice corollary data point from mice, showing that Cal6A3 expression is negatively correlated with ethanol consumption in a two-bottle choice experiment, where mice have the option to drink plain water or water with alcohol added. Okay, our second very interesting signal is on chromosome 13 in the CLIP12 gene. Now, what you see here is a very unusual and potentially anomalous result for a G loss. This is a single very low frequency imputed allele with no correlated signal anywhere around it. This was the same problem that we saw with the chromosome 3 genome-wide significant locus. Now, in the case of the chromosome 3 locus, there are other SNPs in LD with that marker in the 1,000 genomes data that would be expected to show correlated signals and do not, which is why we think that signal is an anomaly. In this case, however, this SNP has no R squared greater than 0.1 with any other marker in the entire 1,000 genomes data set in subjects of European descent. So the lack of correlated signal here is actually as expected given the known population LD structure in Europeans. This gene is a member of the cripple-like zinc finger protein family. These are generally negative regulators of gene expression. And we see very, very consistent model organism data again. Now here, I have to stop and talk for a minute about acute functional tolerance, which is the second phenotype that we measure in the C. elegans experiments. And the reason I want to make sure this is clear is that this is a very important phenotype across species, and it's something that is seen routinely in measures of all species and all behaviors. The paradigm for the worms is shown in red, and that for humans is shown in blue. So in studies of humans, what you do is you take some sort of baseline assessment of performance on a task, and then at T0, the subjects are given a single dose of ethanol, usually an oral challenge. And as that oral challenge begins to be absorbed through the gut, their blood alcohol concentration begins to rise. They're being continuously monitored for performance on the task as their blood alcohol rises. And at some point, some blood alcohol measure, they will drop below an investigator pre-specified threshold for performance on the task. All right, so we mark this as the point at which they lose the capacity to perform the task. Blood alcohol continues to rise. They're monitored throughout this period. At some point, the blood alcohol peaks, and now metabolism takes over from absorption, and blood alcohol begins to fall again. And what is observed for all phenotypes and all species, as far as I know, that have ever been studied, is that you regain the threshold task performance at a higher blood alcohol concentration on the falling curve of the shoulder than you lost it on the rising curve, right? And so this is the definition. It's a decrease in response to alcohol, or a reduction in the impairment of your performance by the alcohol that occurs independently of changes in blood alcohol within a single trial. So this is a very important phenotype, and it's relevant across species. Now, when we do this in worms, obviously we can't give them an oral challenge. So here, we do exactly the same experimental setup that I described to you before. Baseline measurements are taken off of an alcohol plate, and then at T0, the two strains, the mutant and the wild type, are introduced onto the alcohol plate. They begin to absorb alcohol through their cuticles, and their blood alcohol rises, and it gets to a certain point. So we take a measurement at 10 minutes, and at some point, their blood alcohol more or less levels out, although it does continue to rise slightly over time. We then take a second measure at 30 minutes. And to calculate acute functional tolerance, we take the percent of baseline at 30 minutes, which given this figure you would expect to be higher than their percent of baseline at 10 minutes as they develop acute functional tolerance. And we subtract from that their percent of baseline performance at 10 minutes. And that difference is the measure of acute functional tolerance. So here, in this study, we see that while the wild type and CLIF3, which is the ortholog of CLIF12 mutants, are not different for initial sensitivity, and the wild-type animals develop acute functional tolerance as expected, the development of acute functional tolerance is completely ablated in these CLIF3 mutant animals. So this gene in C. elegans is very important for the development of acute functional tolerance in this first trial period. When we look at mouse brain gene expression data, we see that expression is positively correlated with acute functional tolerance. So again, we see beautiful correlation in the behavioral phenotype from nematodes to mammals. If we ablate the expression of this gene, we ablate the development of acute functional tolerance. If we increase expression of this gene, we increase the development of acute functional tolerance. And interestingly, Jill Bettinger and Andrew Davies had already shown in a prior experiment that the obligate binding partner of this gene in both of this gene product in both C. elegans and humans, CTBP1, also has an effect on tolerance. So if you look at this graph here, you can see that while the development of acute functional tolerance is not completely ablated in these CTBP1 mutants, it is definitely attenuated by comparison with the wild type animals. So both of these two gene products, which act together, have an impact on acute functional tolerance, which we think is very, very good support for a role for this gene. The third interesting locus I'm going to tell you about is the RYR3, ryanodine receptor 3 gene on chromosome 15. Biologically, this is a very, very plausible locus. These ryanodine receptors come up again and again and again in discussions of the impact of alcohol on nervous tissue. They are intracellular calcium channels that release calcium from cellular storage, and upregulated calcium-induced calcium release has been strongly implicated in the rewarding properties of exposure to drugs and used substances. This ryanodine receptors are also known to modulate the function of the BK or slow-1 potassium channel, which has been a real focus of interest for the C. elegans group in the VCUR. And they're known to interact with these chloride intracellular channel proteins, which has been a focus of interest for the mouse and Drosophila groups. Unfortunately, although we do see changes in behavioral response to ethanol in 3 different model organisms, the convergence of those specific phenotypes that are affected is not as clean here as it is for the first 2 genes. So for the sake of time, I'm not going to go into detail about them. We do see effects, but we don't see a consistent phenotype yet. And finally, the last locus I'm going to talk about in this study is a long non-coding RNA gene on chromosome 4. Now, replication in human GWAS of alcohol dependence has been extremely poor so far, and our sample is no exception to that rule. This is the one site at which we see some evidence of replication in our follow-up studies. So the initial discovery p-value is 7 times 10 to the minus 7. When we do a joint meta-analysis with the other 3 case control samples, we see this rise to 4 times 10 to the minus 8. So we actually achieve genome-wide significance in the joint analysis. The associated allele is within the mature transcript, so it is potentially functional within the context of a non-coding RNA molecule. And we think that this this particular long non-coding RNA may be binding microRNA genes. Link RNA in general are a set of genes of emerging importance in studies of brain phenotypes. They appear relatively late in evolution. Approximately a third of them are limited to the primate lineage, so there's been a dramatic expansion of the number of such genes in the primate lineage. And in mouse studies, more than half of them are expressed in the brain. And currently, there is interesting and supporting data linking specific link RNAs to schizophrenia, to autism, to a variety of neurological disorders, to other substance abuse disorders, and I think these are going to be something that turn out to be quite important in the brain. Most of them are expressed in very specific regions or cell types or subcellular compartments, which I'll explain why that's important in a moment. So given the specificity of these genes in the primate lineage, and we did look to see whether there was an ortholog of this locus in any of our DCUARC lab species, and there is not. So this is one that does appear to be primate specific. So we can't go back to C. elegans or to mice to really work on this further. So we here turned to post-mortem brain tissue from alcohol dependence cases and controls from the Australian Tissue Resource Centre in Sydney. We have access to nucleus accumbens and prefrontal cortex post-mortem tissue. And so we went to look and see what gene expression for LOC339975 looks like. And when we stratify by clinical status, we see no difference in expression between cases and controls, which I think is actually good given what I'm going to say down here, because this suggests that this is not an ethanol-responsive gene. A long history of drinking does not appear to alter the expression of this gene relative to its expression in controls, at least in these two tissues. On the other hand, when we stratify by genotype, what we see is that carriers of the associated non-reference allele have substantially reduced expression and significantly reduced expression relative to reference allele homozygotes in the nucleus accumbens, although not in the prefrontal cortex. We think this is okay because it's known already that these genes are pretty highly specific in their site of expression, so this may only be relevant to the nucleus accumbens. I'm not going to talk too much today about what we think this gene is doing, but we think we know what it's doing, and experiments to document that are underway, and hopefully I'll be able to talk about that within the next few months. Okay, finally, our most recent and one of our most exciting studies is the China, Oxford, and VCU Experimental Research on Genetic Epidemiology, or CONVERGE, Genome-Wide Association Study of Major Depression, which was undertaken by sparse sequencing rather than array genotyping. This was a massive sampling effort involving 59 hospitals in 45 cities and 21 provinces across eastern and northern China. And I think the most important thing about this study is the way that the sample collection was designed and how the phenotypes were assessed. So a very strong effort was made to reduce heterogeneity and increase genetic risk for depression wherever possible. So all of the samples are female, and this is because male-female genetic correlations for major depression are only about 0.6, which indicates an incompletely overlapping set of genetic risk factors. So we wanted to eliminate the non-overlapping factors in males and focus on females. All the samples are Han Chinese, as previously with the Irish studies, to minimize ethnic and cultural heterogeneity. They had to have 2 or more lifetime episodes because recurrence indexes high genetic loading. They had to have no history of substance abuse to avoid this known comorbidity of uncertain causal direction. The cases had to be over 30 years old to minimize diagnostic conversion to bipolar later, and the controls had to be over 40 years old to minimize late onset major depressive disorder. In the end, we had 5,300 recurrent female hand depression cases and 5,300 middle-aged, never depressed hand female controls for analysis, and replication was undertaken in a further sample of 3,000 hand cases and 3,000 hand controls, both male and female. We identified 2 genome-wide significant signals, both on chromosome 10. The first one is 5' of the C2 and 1 gene, and another very helpful feature of this study was that a substantial phenotypic data, including measures of environmental risk, were collected for these individuals. So it's well known that childhood sexual abuse is the single strongest risk predictor for major depression in women. And data was collected on whether there was a history of sexual abuse in the cases. Because this is such a strong environmental risk factor, we would expect to increase genetic risk if we exclude cases who endorse childhood sexual abuse. So we stripped the analysis down to 4,700 CSA negative cases versus controls, and we see the p-value increase by an order of magnitude. Similarly, when we restrict analysis to the 4,500 cases that satisfy the definition of melancholia, which is a particularly severe form of major depression, versus controls, we see the p-value increase by an order of magnitude again. And the joint discovery and replication analysis sees the p-value increase by 2 orders of magnitude. So we do get support from the replication sample in this locus. The second signal is in the phospholysine phosphohistidine inorganic pyrophosphate phosphatase, or LHP. I did have to practice that for quite a while to be able to spit it out properly. Now in this case, we see no increased signal when we limit the analysis to CSA negative cases or melancholia cases, but in replication we see a very, very strong replication here. So we go from 10 to the minus 8 in discovery to 10 to the minus 12 is the joint reanalysis. And you can see here that the other features of the replication look very good. So this is the discovery odds ratio, 1.16. This is the replication odds ratio, 1.3. For CERT1, this is the odds ratio 0.839 for the discovery and 0.838 for the replication for LHBP. Okay, so all of our data has been contributed to the Psychiatric Genomics Consortium. This is a group undertaking very large sample mega-analyses of GWAS data for psychiatric traits. Multiple members of the Institute are active participants in various working groups, the schizophrenia working group, the alcohol and drug, major depression, CMV, cross disorders, and so on. And then just briefly, several of our active other current projects underway. We're collecting whole exome sequence in the cases from our alcohol dependence sample. There's an extensive literature on the role of rare functional protective alleles, for example, loss of function alleles in aldehyde dehydrogenase genes that lead to buildup of acetaldehyde and facial flushing, which is very aversive and generally is protective against alcohol dependence. But there's been very little study of rare functional risk alleles. In the second study, we're collecting axiom array GWAS data in the Spit for Science sample. This is an approximately 7,500 member cohort of 3 consecutive entering VCU freshman classes, in which we're going to conduct a population GWAS of substance use and other mental health issues, including known exoparameters with genotype in this array. And finally, in ALSPAC, we're performing a GWAS focused on alcohol phenotypes in another 4,500 population subjects. So that's the rundown of our current molecular work and future directions. Thank you very much for your attention. I'm going to turn it over now to Jack.
Actually, it's 20 so 10 needs 10 minutes, 20 minutes.
No, just go ahead and I don't have time.
Middle school.
So, really got at least 25 minutes.
So hold it back, way back down. This is the slide back, slide forward.
Okay, so along the same theme, this is a project looking at a genome-wide association study of anxiety disorders. So everyone has to have a catchy acronym. So this is the Anxiety Neurogenetics Study, or ANGST project. I'm not the originator of that. So the idea here was, instead of looking at genetic risk for individual anxiety disorders, based upon prior work our group and others have done looking at the genetic risk across, in twins, we are actually looking across various anxiety disorder phenotypes and combining association with genetic variants that are shared between those. Even though anxiety disorders all are in the same chapter in DSM, they're quite heterogeneous in terms of their symptomatic presentation. This project was started about 5 years ago, and we originally proposed to have 3 discovery samples totaling about 3,000 cases and 5,000 controls or something, and then use the other samples for replication. But of course, as the PGC helped us understand along the way, that was not going to be nearly enough, so we just combined all of our discovery samples together, and we'll be looking forward to replication in a future time. But we eventually recruited collaboration from 7 worldwide sites with total sample sizes of over 18,000 overall. We had somewhat atypical phenotypic definitions. As I mentioned, we did follow sort of a conventional approach of case control analyses, only as I mentioned, because we are combining across different anxiety disorders, we took a simple-minded approach first in that subjects who had full-blown cases of any of these disorders down here, generalized anxiety disorder, GAD, panic, agoraphobia, social phobia, specific phobia, they would be the cases. And what I did was phenotypically scored each of the subjects in every one of the samples myself. For those who had any significant symptomatology but did not reach diagnostic criteria for any of the disorders, I called them a 1. Full disorders I called a 2, and those who were very, very low or negligent on symptomatology for any particular disorder, we called a 0. And that's of course because clinical cutoffs don't really define who carries risk loci, and those who are highly symptomatic could easily, but classified as controls, could easily contaminate and dilute the comparisons. So we took the 2s versus the 0s, any anxiety versus supernormal controls. But then we did something else that takes a little more thought and is consistent with the way we do things here, and that is we constructed a factor analysis. This is just a phenotypic factor analysis. Most of the samples didn't have twin data for us to be able to have genetically informed modeling. But what this allows us to do is look at the covariance between the individual disorders and score that into a single factor model from which we extracted the factor score for every individual in the samples. And then we use that as a quantitative phenotype as in a second GWAS analysis. We did not have access to the individual level data. For the Angst Consortium, you are not required to share all your data, and so some of the sites really were not allowed to do that, or not very easily. So these analyses were done within each sample, and then we conducted a fixed meta-analysis with 1,000 genome imputed SNPs for all the samples. And we conducted the usual SNP and gene-based tests, but because we didn't have a replication sample, we conducted some interesting internal cross-validations where you drop one sample from the analysis, conduct your GWAS again, and look at the correlation between the findings to see if they are higher than you would expect by chance. So this is the case control GWAS findings of Manhattan plot, and similarly for the factor score. And we have genome-wide significant hits, one for each of those analyses, but in different chromosomes, even though they, in principle, these should tap into some overlapping phenotype. And certainly when you just calculate the correlations between the phenotypes and between the GWAS association hits, there's extremely large correlation, but the highest values of significance were not the same in both samples. So for the case control, it was as on chromosome 3 in this SNP. These are both fairly high minor allele frequency SNPs, so common alleles. There's a fairly wide range of positions in one versus the other. I'll show you that in a minute based upon LD. The effect sizes are around the range that you would expect, fairly low. For the case control, this is an odds ratio, and this is a beta value. Both p-values, as I mentioned, were genome-wide significant, as were the false discovery rates. In all the different samples, the directions were in exactly the same, the hits were in exactly the same direction. And if you look at this SNP and its association with this phenotype, there are nominally significant in the cross analysis. The first discovery was on chromosome 3 for the case control study. This is the regional plot. This particular locus is actually a uncharacterized non-coding RNA of the type that Brien was telling you earlier. There were 3 SNPs that were genome-wide significant in that region, and as you can see by the forest plots, the signals were quite consistent across all the different samples. So no one knows anything about this gene. We certainly don't either. For the factor score, our most significant region was on chromosome 2, where there are a number of genes actually very high LD, which is why these signals were relatively flat, because the LD was something that was greater than 0.9 across a very large region. But again, the signals are very consistent across the different samples. We know a little bit about some of the genes in this region. This PREPL gene has to do with the maturation and degradation of peptide hormones and neuropeptides, which is at least somewhat related to brain and stress. And these other 2 are actually related to a Mendelian disorder of amino acid transporter malfunction that leads to a cystitis kind of syndrome in children. This one particular gene, the CAM-KMT, is related to calcium signaling, which is at least vaguely consistent with what we see for other psychiatric disorders from GWAS and the PGC. And this is not mine, so I guess we're done.
Question for Brien. Your top 6 of chromosome 10, p-values for PGC range of detection for the same regions.
Tim has just finished that analysis, right? We did not. Yeah.
So my question is, do you think that the converged design and its focus on harm and its rigorous control is a better check on potential heterogeneity on PGC?
So there's been a lot of debate and discussion about questions like that. So in the schizophrenia analysis, this is now 2.5 years ago, there was a lengthy debate about whether to include East Asian samples in the primary PGC meta-analysis of schizophrenia. A lot of people argued against it, arguing that we needed to maintain genetic homogeneity and analyze just the European samples first. But type 2 diabetes studies, which are a couple of years ahead of everything else, indicated that by and large, the same loci yield signals in samples of European descent and samples of East Asian descent for type 2 diabetes. We can talk for a long time about the specific issues in type 2 that might make that the case. It's not necessarily going to be the case for other disorders, but the decision was made in the schizophrenia analysis to jointly analyze East Asian and European samples together. In the converge data, one of the things that we do see is that this is a region where East Asian LD patterns and European LD patterns are quite different, and the allele frequencies are quite different. So the allele is common in East Asia, and it's pretty low frequency in Europeans. So this may just be a power issue. We really don't have the power to detect that signal in European samples.
Use the converge results to estimate Chinese polygenic risk in major depression in Caucasian samples.
Yes, can I ask that question to someone else?
I think that they did, and they are significant. Not at all, but they are significant.
is something there. It's worth
Yeah.
it.
Given that we need pretty monstrous sample sizes to do a lot of this research and actually hope for discovering very large effect sizes.
Right.
Pray, not even hope, we're beyond hope.
Well, I think we'd be happy to find things that are significant, even if they're not large. I'd be suspicious if we found anything that was large.
Okay, given that we need substantial effect sizes, or substantial sample sizes in order to find minuscule effect sizes, and given that this is going to, in order to collect such data, we're probably going to exceed any standard NIH level grant. And the problems with co-authorship and crediting individual investigators, what is the proposal going forward in order to keep collecting such data and staying in the game?
I think that's a very good question. I'm not sure that I have a full answer for you. There was a proposal initially in the PGC to undertake sample collection directly and try to directly increase the sample size available, and IMH said no to that. They were not willing to fund that. In general, investigator and groups of investigator response to that problem has been to form consortia. So this is why you see this proliferation of consortia across the GWAS literature. And in some cases, data for height, for example, you can get several 100,000 people available for that phenotype, at least the last time I heard. Ultimately, I think we are going to have to continue to add samples. I think we are going to have to continue to collect additional samples and to collect this level of data on whether it's by array genotyping or if sequencing becomes cheap enough that you can sequence the whole genome of every individual we collect, which will yield a nicely consistent, non-imputed, directly observed set of genotypes. I don't know, but it's going to be very expensive. And the same, I'm sure you know, the same problems affect the study of rare variation, because although the effect sizes are expected to be big, the allele frequencies are very tiny. So you need exactly the same size sample to study rare variants that you need to study common variants. So we're going to have to sequence 80,000 or 100,000 people before we get good rare variant traction in schizophrenia, for example, and probably more for depression and alcohol. So we need cheap technology and bigger samples.
What's all it takes is one private individual in 23 years of reproduce that.
And I think certainly collaborations with groups like the Kaiser Permanente in Northern California and 23andMe have yielded substantial numbers of samples that can be added in. On the other hand, the data restrictions are often as bad as for pharma data if you're dealing with healthcare or private company data. So they're willing to work with you, but you generally cannot access the raw data, which is not ideal.
The polygenic heritability of depression from 23andMe, which is a single tick box, is about 7%. So it's there, but it's not very heritable, which would make sense. It's a very noisy phenotype.
So probably when we'll have medical records and everybody will do sequence genotype, we can find out a lot more great from the
Well, I think so. Some of you were probably either in Toronto or on the recent PGC call when Nancy Cox described what they're doing at Vanderbilt at the moment, which is they're collecting, they were initially doing exome sequencing. I think they got to about 45,000, 50,000 people with X. So these are individuals who are coming through the hospital system for routine treatment. They get diagnosed with some condition. They agree to participate. Their electronic medical records are available anonymously, and their biological samples are, in this case, sequenced. And now they're moving to whole genome sequence. They expect they're going to have 200,000 genomes sequenced by a year from now. And one of the things that's been very powerful there is identifying relationships between genes that may predispose to two very different phenotypes. So if, for example, you have a signal from schizophrenia, I'm trying to remember what the actual Do you remember what the thing she discussed was? There's an interesting conjunction of data where they went looking at other phenotypic presentations in carriers of an allele that had been associated with something, Crohn's or type 2, I'm not sure what, and found a second set of association, a phenotypic level association centered on that gene for a very different phenotype. But you can get access to very large samples this way.
And of course, there's the UK biobank, which will be available in a couple of years' time as well.
How big is that? Is that 100,000?
100,000 plus. So, and they're assessing everything.
Right.
Plus, I think, MRIs and
Also you have the Scandinavian countries, you know, satellites and
Emphasizing. I guess to follow up then, how do we as an institute get involved, like, beyond 50 authorship?
Well, you can become the director or senior analyst for a PGC working group. That's a great way to get a good publication. If you contribute your data, you're going to be part of the consortium authorship list. I think there's really no way around that. So much work has been done just in the PGC2 schizophrenia data set, however, that even if you're not the first author on a consortium paper, there's plenty of work to be done. And, you know, I think things like we're trying to do with the recurrence risk data, that's an area that nobody's really explored. And it's very contentious. Most people you talk to expect that family members are going to have lower polygenic risk scores because they expect a portion of their risk to be made up of rare variants diseases, segregated in there. But I think that assumes that risk for schizophrenia is like a unitary value and everybody has essentially the same risk. And in fact, the recurrence data says people in families actually have higher risk than your standard population case. So I think those sorts of analyses can still be done and can support other work that you might want to do, like doing deep sequencing in a relatively small but ethnically very homogeneous group of families. Right? And something like the evidence from the PAC7 duplication really supports our contention that if you sequence within a limited ethnic group, say within a population, you dramatically increase the power that you have to detect specific alleles, which is the real holy grail of this, right? Because we haven't done that really in GLS yet, and it's proved very, very challenging to do the sequencing when the sample sizes aren't large.
We also, of course, have access to two samples directly. One is the Speak for Science project, which we'll be writing a renewal for in a year that we may hope to expand, and Converge. We have an application into the Wellcome Trust to expand to 48,000 cases and controls, which just a few days ago passed the next major level, so we're going for a reverse site visit there in the middle of December. We have preliminary efforts to try to gather a large sample of subjects with alcohol use disorder, which we're in discussion with the NIAAA Institute. So there's several other more ongoing efforts.
I think obviously sampling outside of Europe or North America is dramatically less expensive. So working in China, there are limitations. Samples can't leave China by government decree. You often have to negotiate what portion of the data can leave China. But in general, you can generate very, very large samples, big populations. It's pretty genetically homogeneous. It's not as homogeneous as a single as Iceland or Ireland is. But the Han Chinese population does give you some reduction in it. It's inexpensive to sample. And if, you know, somebody who really wanted to kind of move into a completely new area, sampling in Africa is almost not happening, right? And the representation of people of African descent and people of Native American descent in these data sets is virtually nil. The genetics are harder in Africa, but there's also a lot of work to be done there. And sampling costs are, you know, maybe 1% to 5% of what they are elsewhere. So I think that's another possible answer to your question, is to think creatively about who to sample and who to include. All right, thank you.